My name is Dr. Stephen Thompson. I'm from the University of South Carolina, and I'd like to share with you some activities that you can use to help learners understand cellular respiration occurring in plants. It's one of the biggest misconceptions that students hold, that plants do not engage in respiration. So, the first activity is pretty simple. It involves pea seeds, the plant's peas that we all love to eat. Involves taking pea seeds and taking pea seeds that you would buy in a local hardware store and germinating those. So yesterday I inserted some water in this container. The seeds have been germinating overnight, and what we're going to do is simply measure the level of gases that are being created by each of these two containers. Because we're talking about respiration, the two gases we're interested in are oxygen and carbon dioxide. So to conduct the activity, usually what I ask students to do is consider what kinds of gases would be produced by each of the dormant and then the germinating seeds. And then we use a device to measure the carbon dioxide reading in each of the containers. So I'll go ahead and do that. So after I take the readings of the CO2 and the oxygen, what students find is that the container that has germinating pea seeds has higher CO2 and lower oxygen levels than the container that has dormant pea seeds. So one of the big ideas that I'm trying to get students to understand is that germinating seeds do something that dormant seeds do not. Germinating seeds are basically baby plants. They don't have leaves. Because they don't have leaves, they're not engaging in cellular respiration. So when students encounter high levels of CO2 or carbon dioxide in this container and low levels of oxygen, it's evidence that these seeds are engaging in cellular respiration and these seeds not so much. And so it's one of the ways I try to help students understand that plants respire just like animals, just like insects, just like fish. Another way that I show this is by using a plant. Any plant will do, and the second experiment just involves taking any plant that's alive and thriving, and just like in earlier videos where we measured the oxygen production in a plant, we're going to cover up this plant, we're going to seal the plant part pretty tight, and what I want to do this time, instead of placing the plant in sunlight, I'm going to put this plant in a dark location for 24 hours. Any dark location works, a box, a closet. And then after 24 hours, we're going to measure the gases that are inside this plastic bag. And what I hope students will see is that the container will have high levels of carbon dioxide and low levels of oxygen. It's the evidence that the plant is engaging in respiration. We've eliminated sunlight, which means that photosynthesis is not happening. And so the only process that is taking place that we're interested in is the cellular respiration that plants engage in. And that high CO2 is evidence that it's breathing out CO2 and taking in oxygen.